New research from Washington University School of Medicine is shedding exciting light on one of nature's most finely tuned reproductive mechanisms: how mammalian sperm detect and respond to heat. This new understanding not only clarifies one of the great mysteries of reproductive biology, but also opens promising new directions for fertility treatment and birth control alike. What makes this discovery especially compelling is the elegance of its simplicity. Temperature acts as a switch. In that switch, controlled by a protein called Spur, may hold the key to both unlocking fertility challenges and designing targeted contraceptive strategies with fewer side effects. Sperm cells, by design, are incredibly sensitive to their environment. Unlike many other cells in the human body, they operate best below the body's core temperature, which is precisely why testicles are housed in the scrotum, external to the body. This spatial arrangement is no accident of evolution. It serves to keep sperm cells dormant and stable until the precise moment they enter the warmer environment of the female reproductive tract. At that point, everything changes. The temperature increase above 38 degrees Celsius acts as a biological signal, flipping a molecular switch that activates a protein on the sperm's surface called spur. The result is a dramatic change in movement. Calm, slow swimming turns into vigorous, whip-like thrashing, a behavior critical for the sperm's final mission, piercing the egg and achieving fertilization. Until now, scientists believed that spur responded primarily to chemical cues like progesterone or changes in pH. But this study reveals that temperature may be the true master regulator, providing a cleaner, more reliable cue. This new insight makes perfect evolutionary sense. Progesterone varies between species and even within individuals, whereas temperature remains a consistent, biologically meaningful marker across all mammals. This thermal activation mechanism also explains the elaborate anatomical adaptations seen in mammals to maintain cooler testicular environments, dolphins cooling their blood through dorsal fins or elephants using their ears. It's a remarkable demonstration of how evolution sculpts physiology to protect even the smallest and most vulnerable components of life's continuity. What's more, this temperature-sensitive protein offers a highly specific target for future medical applications. Because spur is found only in sperm, any drug designed to influence it is likely to have minimal impact elsewhere in the body, something that's critically important in both fertility and contraceptive research. Imagine a male contraceptive that doesn't rely on hormones or affect testosterone, libido, or mood. If researchers can develop a way to prematurely activate spur, perhaps through a slight and temporary warming effect, they could exhaust the sperm's energy before it even reaches the egg, rendering it harmless in a safe, non-disruptive manner. This represents a major leap forward in the long search for effective, reversible, and side effect-free male birth control. On the other side of the reproductive equation, this study also provides important new insights into female fertility, particularly how it changes with age. By analyzing ovarian tissue across decades of life, the researchers identified that different regions of the ovary age at different rates, with significant functional decline appearing even before menopause. Notably, the support systems surrounding eggs, rather than the eggs themselves, seem to break down first. Granulosa and stromal cells, critical to maintaining a healthy egg environment, show signs of degeneration, while fibroblasts begin to lay down stiffening, fibrotic tissue that interferes with reproductive function. Here again, science offers hope by identifying that one of the culprits is reduced TGF-beta signaling, a pathway involved in tissue repair and inflammation. Future therapies might focus on boosting this mechanism to maintain a more youthful, supportive ovarian environment. The idea that fertility loss is not solely about egg depletion, but also about the breakdown of surrounding infrastructure is an empowering one. It suggests that fertility might be preserved longer than previously thought, not by delaying the inevitable, but by repairing and reinforcing what keeps the eggs viable. Together, these discoveries represent a shift in how we understand reproductive health. Rather than treating fertility as a fixed clock or a purely hormonal issue, We can now approach it with the precision of molecular science, targeting specific proteins, cellular behaviors, and even heat itself. For men, it means smarter contraceptives and better understanding of infertility. For women, it may mean preserving fertility further into midlife and potentially preventing some of the early decline associated with ovarian aging. In an era where both partners increasingly share responsibility in family planning and fertility journeys, the dual insights of this research offer balance and shared hope. It's not just about fixing what's broken, but about enhancing what nature already does so well, with a little help from temperature, time, and human ingenuity.